Hello everyone. My name is Larsal Obeidi, and today I will be presenting a study titled Gut Microbiota Dysbiosis and Vitiligo, the role of hippuric acid in oxidative stress. This presentation is a part of my work for a microbiome classes, and it explores how imbalance in gut microbiome may contribute to skin autoimmunity, especially vitiligo, through a microbial metabolite called hippuric acid. I will be walking you through a background, methodology, result, and implication of this study, and we will finish with some discussion questions. So let's get started. To begin, the bottom line takeaway of this study is that gut microbiota dysbiosis may play a causal role in vitiligo by promoting oxidative stress, while the connection between the gut health and skin disorders has been suggested before. The exact mechanism wasn't clear. So, this study aimed to fill the gap by identifying a specific microbial metabolite, puric acid, as a key link between gut dysbiosis and oxidative damage in vitiligo. The researchers used an integrated approach involving clinic data, animal model, and multi-omics technique used to demonstrate that sulfuric acid levels were elevated in both vitiligo patients and mouse models with dysbiosis. This work provides strong evidence for good skin access in vitiligo and open the door for new therapeutic approaches targeting the microbiome. Before we diving into the study itself, it's important to understand the biological context. The gut microbiota is known to influence immune homeostasis and oxidative stress throughout the body. Meanwhile, vitiligo is a chronic autoimmune disease where oxidative stress plays a central role in damaging melanocytes, the cells that produce pigments in the skin. Although recent studies have hinted at a gut-skin connection, particularly in autoimmune conditions, the precise mechanisms involved in vitiligo have not been clearly defined. This sets the stage of the current study, which investigates how microbial metabolites, particularly hippuric acid, may mediate these gut-skin connections and drive disease pathology. As the researchers note in their abstract, the complex integration between abnormal accumulation of ROS, which is the reactive oxygen species, in the skin and gut microbiota has remained unclear. So this directly underscores the knowledge gap they aim to address, namely determining how gut-derived factors like uric acid may be driving this process. Now that we have covered the background, let's talk about why this research is needed. Although the gut-skin axis is being increasingly recognized, we still don't have a clear understanding of how gut dysbiosis may contribute specifically to vitiligo. Vitiligo is largely driven by oxidative stress, but what actually triggered that stress at a molecular level hasn't been fully defined, especially in connection with the gut microbiota. Current treatments mostly focus on topicals like steroid or immunosuppression, but these approaches don't target the root cause. So, this study addresses an important gap, exploring whether microbial metabolites, particularly uric acid, could be the missing link and potentially a target for future therapy. So, this brings us to the core research question. The researchers wanted to know whether gut microbiota dysbiosis contributes to vitiligo progression by targeting oxidative stress. Especially, they ask whether a gut-derived metabolite, uric acid, plays a central role in this process. They also aim to determine whether this link could help explain how gut health influences autoimmune skin conditions like vitiligo. Answering this question could shed light on previously undefined mechanisms and potentially identify new therapeutic targets. So this study used the integrative multi-omic approach comparing both clinical and animal models. The researchers first collected stool and blood samples from vitiligo patients and healthy control to assess gut microbiota compositions and serum metabolite levels. They then used a mouse model of vitiligo, transferring gut microbiota from patients to mice to see whether dysbiosis could induce oxidative stress and depigmentation. They applied 16 sRNA sequencing to analyze the microbiota and metabolomic to identify differentially abundant serum metabolite, with uric acid emerging as a key candidate. To test causality, they administered uric acid directly and monitored skin responses and oxidative markers. So, RNA sequence and histology were used to further investigate skin gene expression and damage pathway. In this part of the study, the researchers used a variety of molecular and analytical tools. First, they performed 16 sRNA sequences to determine how the gut microbiota composition differed between vitiligo patient and healthy control. Next, they use serum metabolite to identify changes in like the changes in circulating metabolites, focusing on uric acid, which was significantly elevated in vitiligo. So the RNA sequences was used to examine gene expression patterns in the skin, especially gene involved in oxidative stress and immunoregulation. So to confirm tissue level effects, they use histological staining and Rose assays to show melanocyte damage and elevated oxidative stress. Finally, by transplanting microbiota from patient mice, They demonstrated that gut dysbiosis alone could target vitiligo like features. So here the figure one, the summaries an RNA sequence analysis of lesional and non-lesional skin samples in vitiligo patients. Panel A shows the study using publicly available data from GEO. Panel B demonstrates a principal component analysis where lesions and non-lesion samples clearly separated, indicating distinct transcriptional profiles. Panel C shows the number of genes significantly up or down regulated. Panel D displays a heat map of DEG with many involved in oxidative stress and immunoregulation. So finally, panel E shows gene ontology enrichment, highlighting pathways related to ROS production and inflammatory responses. These results suggest that oxidative stress is transcriptionally active in vitiligo lesions and sets the stage for connecting microbiota drive metabolites like puric acid to skin level dysfunction. So in summary, the RNA sequences data from lesional skin confirm increased oxidative stress and immunoactivations in vitiligo. When this is paired with findings from the microbiome and metabolic arms of the study, we get a clear picture. Like gut dysbiosis in vitiligo patients lead to an increased production of puric acid, a metabolite capable to targeting oxidative stress in the skin. This creates a mechanistic link between the gut microbiota and melanocyte destruction. Helping explain how gut health may influence skin autoimmunity. This supports the growing evidence of the gut's skin axis and also points to potential new therapeutic directions like probiotic or metabolite based intervention. So in the conclusion, this study presents strong evidence that the gut microbiota dysbiosis contribute to vitiligo progression through the metabolite puric acid by linking microbial imbalance to oxidative stress in the skin. The researcher established complete gut skin access mechanisms. So to use of multi-omic tools combining microbiome profiling, metabolomics, and transcriptomics offers a puric understanding of disease. Most importantly, this opens the door for a new treatment strategy aimed at restoring microbial balance and reducing systemic oxidative stress in autoimmune skin conditions like vitiligo. So, to wrap up, I like to open the floor to discuss this discussion with a few questions. How strong do you think the evidence is that hippuric acid drives oxidative stress in vitiligo? And what are your thoughts on the value and challenge of combining microbiome metabolomics and transcriptomic data? And could you influence uric acid levels in a clinical setting through diet or supplementation? And can you think of other autoimmune or inflammatory disease that may share this gut skin mechanism? Do you think we should be targeting specific microbes or just the harmful metabolite they produce? So finally, these are the reference I used, including the awesome article we just explored together and some supporting reads about the microbiome and skin health. Thank you so much for listening. I hope this talk gave you something new to think about, or at least made you look at your gut a little differently. It may be a short presentation, but give us a lot of new knowledge. I would love to hear your thoughts or answer any questions here. Thank you.